In this video, we will discuss about this ALDH4A1 ability to import pyruvate functions in mitochondria depends on its interaction with this MPC1 and MPC2. So that's what we discussed, that it's not the catalytic activity of ALDH4A1 that determines its pyruvate import ability, but its interaction with this MPC1 and MPC2 that determine its pyruvate import ability. So this is what we will be concluding at the end of this results that are presented in this figure. So we'll start. First, they wanted to check that ALDH4A1 interacts with this MPC1 and MPC2 or not, and for that they did the immunoprecipitation assay, and they are able to find out that in the case where they knock down SH, this ALDH4A1, and they are able to see less amount of this MPC1 when they immunoprecipitate MPC2. When they immunoprecipitate MPC2, they are able to see this MPC1 also interacting, indicating that this MPC1 and MPC2 are interacting to each other. And they did the similar immunoprecipitant assay with MPC1, and in this case they are able to see that MPC2 is also present in the case of this control, and also in the case of this ALDH4A1 is knocked down, indicating so far they have indicate or proved that this MPC1 and MPC2 are interacting to each other and form a complex. Now they wanted to check the ALDH4A1 will interact with this complex or not. So for that, what they did is they immunoprecipitate this ALDH4A1, and in that case they are able to see that these MPC1 signals are clearly evident, and these things they have did it with this MPC2 also, and they are able to see that MPC2 signals are also evident, indicating that this ALDH4A1 is interacting and making a complex with this MPC1 and MPC2. And these things, and thereafter they checked the effect of this ALDH4A1 on the pyruvate imports or not, and they are able to see that in the case when there is and they are able to see this ALDH4A1's ability or when this pyruvate. So let me just describe about this panel E. So in this case the MPC1 and MPC2 are conjugated with their specific fluorescence, and they are able to see if they use the pyruvate whether it will increase the complex formations or signals or not. So in the case where ALDH4A1 is knocked down, and when they don't use the pyruvate, they are able to see a low amount of signals. Both in the case where pyruvates are used or pyruvate are not used, but when they used this ALDH4A1, when they reintroduce this wild type ALDH4A1, and when they don't introduce the pyruvate, they are able to see less signals, indicating that less complex formations are there. But when they induce the pyruvate, they are able to see more signals, indicating that the this MPC1, MPC2, ALDH4A1 complex formations are high, and thereafter they validate this thing using the immunoprecipitant assay, where when they knock down ALDH4A1, they are able to see a decreased amount of MPC2 formations, and but when they use they see less amount of MPC2, indicating that ALDH4A1 knockdown cause this or affect somehow this MPC2 interactions. But when they use this reintroduce the wild type ALDH4A1, they are able to see again the MPC2 signals. So this further confirms the finding of the pyruvate results, or further substantiate the finding of the pyruvate results that ALDH4 helps in the complex formation of MPC1 and MPC2, thereby helps in the pyruvate import. Now they wanted to check whether the mutated ALDH4A1 also has the similar ability or not. So they did they used two different kind of mutations. First they mutated around 182 to 199 base pair, and second they mutated around 553 to 564 base pair, and then check whether it has any impact on this MPC1, MPC2, ALDH4A1 complex formations. And what they are able to find out is that when they use this first mutation, that is mutations from 182 to 189, in that case they are able to see see this MP in the full length ALDH4A1, they are able to see this MPC1's formations, and that can be seen here. But when they use the first mutated, that is around 182 to 189, they are not able to see this MPC1 signals. But when they use the ALDH4A1 second mutated, that is around 553 to 564, they are able to see the MPC1 signals again, indicating that the mutations at around this 182 to 189 affect the complex formations of ALDH4, MPC1 and MPC2. And these things they have validated using the MPC2 signals also, and here they also find the similar results that MPC2s are absent in the first mutations but they are present in the second mutations. So thereafter what it is, they check this panel J is about to check these 4A1 formations and the MPC1 and MPC2 presence within the cells is a Western blot. So here we can see that in the full length they are able to see the signals for 4A1, and in the case of mutated ALDH4A1 they are still able to see 4A1 signals, and MPC1 and MPC2 signals are also present. And for this 554 to 564 also they are able to see all the signals, indicating that this is immunoprecipitant. In the immunoprecipitant assay, the ALDH4A1 is not able to form complex, whereas the MPC1 and MPC2 are present within the cells. Next, the ALDH4 functions is to convert proline into glutamate from this GSA. So they wanted to check whether proline's amount goes down in these mutations or not. So in the case where ALDH4A1 is knocked down, they are able to see a increase in the prolines, because the prolines consumptions are not been as good as in the control case where ALDH4A1 is present. But when they use the full length ALDH4A1, they are able to see a decrease in the proline formations. And similarly for both the mutant, also they are able to see a decrease in the proline formations. And so this and thereafter so this indicates that even though there is a mutations occurs, it does not affect the ALDH4A1 functions to convert prolines to glutamate. Now they want to check the pyruvate uptake of these different mutants, and they can see that in the case of ALDH4A1, the first mutant, that is around 182 to 189, they are not able to see a regaining of this pyruvate uptake, whereas in the case of second mutant they are able to see the regaining of the pyruvate uptake, which is comparable to the control, which is comparable to the wild type ALDH4A1 reintroductions. So this indicates that the interactions, it's not the catalytic activity of this ALDH4A1 that plays a role towards its pyruvate import, whereas its ability to form a complex with MPC1 and MPC2 that affects this ALDH4A1 ability to import pyruvate in mitochondria. This is how they come to conclusions that ALDH4 ability to import pyruvate depends on its interaction with MPC1 and MPC2 complex and not depends on its catalytic activity. That's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.